So I'd like to welcome everybody to Chapter 12 as we talk about DNA, and today we're going to talk about mutations and what can go wrong. So the first thing we're going to talk about are DNA mutations. First of all, DNA mutations is a mistake or a change in one or more nucleotides. So we're just talking about a very small piece of the DNA, so we're just changing one or two bases or base pairs. Okay. For humans, they occur in about 1 in every 100 million bases, so it's not too common, but they do have some huge effects. And hopefully our mutations aren't permanent. We have proteins that double check when we go through replication to make sure that we don't make a mistake when we replicate our DNA. But carcinogens, things like being out in the sun or asbestos or other hazards that we come in contact with, permanently change our DNA, and those are not able to be fixed. Now we have a couple of different types of DNA mutations. The first one is a point mutation, and just like what it sounds like, a point mutation is where one base is changed. So that's the equivalent of changing one letter in our sentence. So the sentence that we're going to use a lot today is the fat cat ate the wee rat. And in a point mutation, notice that the C in cat is just changed to an H. We're just changing one letter. Now what that happens is, or how that, the consequence of that is that we change, that codon changes, which could change the amino acid. Depending on the amino acid, if it's one of those amino acids that has multiple codons, then we will never see an effect of that. We could have one amino acid that gets changed, which may alter the function of that protein just a little bit, or in some bad cases, we could actually have this point mutation causes a premature stop codon, so our protein will never be able to work, or in also some rare cases, that stop codon gets mutated to another codon and our protein never stops. So one type of mutation, that point mutation, where we are just changing one base, one nucleotide. The other type of DNA mutation is a frame shift mutation. Now we read a codon at a time, and a frame shift mutation is where that codon, that reading frame, changes. So in this case, we either going to have one or more bases that are either inserted or deleted out of our DNA. So here's our sentence again, and in this case we are going to take out the letter T in cat, and every letter gets shifted one base to the left, and so we're still reading in our codons, but now all of our codons look different. So the consequence of this is that from the point of the mutation on, all of the codons are off. They're all weird. So the protein could be a lot shorter. It could be a big jumbled mess. Depending on where the mutation happened, we may never see our start codon. Things just don't work right with frame shift mutations. Okay, so like I said, every amino acid after the mutation is different, and that can have some drastic effects. Now, so those are two DNA mutations, a point mutation or a frame shift mutation. We can also have chromosomal mutations. Now, chromosomal mutations, it's where we see a huge chunk of our DNA, whole genes are mutated. So this usually happens during crossing over in meiosis, where the genes are supposed to swap spots and something just happens to them. It will change the phenotype and the genotype of the individual because they may not have the correct letters anymore. And of course, if they don't have the right genotype, that will change their phenotype. And if it is during meiosis, of course, like we know, it's not going to affect you, but it's going to affect your kids because meiosis is producing those gametes or those sex cells. If it is an essential section of the DNA that's required, like how to do mitosis or how to make a heart or how to make muscle cells, then the mutation will actually cause the organism to abort the offspring before they're born or to have a miscarriage. So we have a couple different types of chromosomal mutations. The first one is a deletion. Just like a frame shift mutation in DNA, chromosomal mutation, deletion, we have a whole section or whole genes that are taken out of our DNA. These are permanently lost because now they're not attached to where they need to be. This is a massive frame shift mutation, and we're going to lack our DNA from the normal chromosome. So if this is our chromosome and these are our genes, we're just going to take out the word cat. So the fat ate the wee rat. Notice this is a little different than a deletion in DNA where we're talking about one. Here we're talking about the whole gene being gone. And here's another example. A, B, C, D, E, F, G, H, I. If all of a sudden D, E, F, G are deleted out because they don't cross over correctly, now we have a much shorter chromosome than we had before. Another type of chromosomal mutation is an insertion. Just like it says, we are adding a section of the DNA. This is usually because as they go to cross over, one gene doesn't leave. The second gene comes in, and now we have a duplication or a multiple genes on the same chromosome. So we're adding extra genes. It can cause, if our replication process doesn't work right, we can actually replicate a section of the DNA more than once. That's what happens in Huntington's disease, where you have the same section of DNA that's multiplied over and over and over again, and that causes you to have Huntington's. So you have your normal chromosome plus extra DNA. So here's our sentence, and I'm just going to add in this extra DNA that came from somewhere. So now my chromosome is longer, and of course when I go to find my gene to do protein synthesis, I'm not able to go to the right spot because there's something else that's not supposed to be there. And here's a replication. I have A, B, C, D, and then C and D got copied again, or they were from the other chromosome and they got added into this one, and so now my chromosome is off kilter. Okay, so that's an addition. That's a chromosomal insertion. Another type of chromosomal mutation is an inversion. An inversion is to invert is to swap around. So in an inversion, this whole section of DNA is actually reversed. Instead of moving the way it was supposed to move when it crossed over, the DNA got flipped around in that process, and when it reattached to the wrong spot. So the normal chromosome is unaltered. It has all the same genes that it's supposed to have. It's just that they're in the wrong order. This is may not be viable depending on the genes. If it's genes that we don't really use that often or aren't that critical, then it's just fine. Inversions also allow advantageous characteristics because now genes are in a different order, which means you might do protein synthesis in a different order. If they're in a line of genes, you could make your proteins in a different order, and that would allow you to do things that you maybe weren't able to do before. Okay, so that's an inversion. We can see here we have our DEF just inverts. They're still there. They're still in the same order. But if your body was always going to make protein C, and then it goes to the next one and the next one, and so on, by switching those around, you now make proteins in a different order, and that may help organisms live. Once again, it just depends on which genes are involved. Okay, the last kind of chromosomal mutation is called a translocation. Trans means across, so we are going across chromosomes this one. This is where part of our chromosome breaks off and is actually added to a different chromosome. This is usually lethal, which means that if it happens, the organism does not survive. So in this case, see our translocation, the J and the K broke off of this chromosome and actually got added to the bottom of this chromosome, and it is now on right after I, and then the second chromosome here is missing its J and K. So we breaking off of one chromosome and it's moving on to another chromosome is a translocation. It's moving across chromosomes, and those are mostly lethal. Okay. So we have 4 main chromosomal mutations. We have an addition, a deletion, an inversion, and a translocation. Now the last thing we're going to talk about is a mutation, but it's not a change in our DNA like the last ones were, and these are called non-disjunctions. We're going to have to go back and think about meiosis when we talk about non-disjunction. This is where our homologous chromosomes fail to separate during meiosis. So non-disjunction is the failure of our homologous chromosomes to separate during meiosis. So remember, during meiosis, our homologous chromosomes came together during prophase, and during anaphase 1, our homologous chromosomes were supposed to separate. During non-disjunction, the spindle fibers don't work very well. Your centromeres are really, really sticky, and they keep the homologous chromosomes won't separate. Or what could even happen is that then in anaphase 2, your sister chromatids can't separate. So if you have monosomy, you only have one copy. So as your homologous chromosomes separated, they split just fine, but then when your sisters went to split, one cell ended up with 2 copies, the other cell ended up with 0. Then as a kid, you end up with only getting 1 copy from a parent because the other parent gave you 0. So monosomy, you only have 1 copy of your chromosome. An example of this would be girls that instead of being XX are XO. Many of them die before they are even born. Trisomy, this is where you have 3 copies. So on the flip side, your sisters did not split apart. You happen to receive 2 copies from one parent, your single copy from your other parent like you were supposed to, and now you end up with 3 copies of a chromosome instead of only 2. The most common one of this is Down syndrome. That's a trisomy 21. A couple others is Edwards syndrome, trisomy 18. People with Edwards syndrome only live till about the age of 2 to 4 months. And Patau syndrome is trisomy 13. The majority of people there, kids that are born with Patau syndrome die their first month after they're born, and then those that are able to survive that first month usually only make it about another 6 months. So Down syndrome is the one that we can handle the best. Any other type of trisomy or even monosomies, humans cannot handle that, and we usually end up dying either before we were born or shortly after birth. So we've talked about chromosome mutations. We have insertions, deletions, inversions, and translocations. Those are mistakes happening to the chromosome. We have non-disjunction, which is where the homologous chromosomes or the sister chromatids then fail to separate the way they're supposed to during meiosis, resulting in either having a monosomy, which means you only have 1 copy, or a trisomy where you have 3 copies of a chromosome. Remember, humans, we like to have 2. And then we also talked about DNA mutations, where we can either have a point shift mutation, so we're swapping out 1 letter, or we end up with a frame mutation, a frame shift mutation, which is either an addition or a deletion of 1 or 2 nucleotides. And then that should wrap up our discussion about DNA and mutations.